Start again. Okay, recording progress. So we're going to talk about species today. That's what we're going to talk about. This is all review. You guys can watch the other videos. We're going to talk about species, and we're going to talk about describing species. So to start with, if PowerPoint will let me. So we talked about ICZN. So when you were describing a species, you are working within ICZN. And we talked about this last week. We talked about species. We talked about genus, family, order, class, and phylum. And ICZN is only here. But if we're describing a species, we're down here. So we are within ICZN, right? Okay. So ICZN, we have to follow ICZN rules. ICZN has lots of rules for species and for species names and for genera. And this is what I want to talk about in the next few classes. So let's start with something really simple. Describing species. This is important. A species is a hypothesis. A species is an idea, right? In other words, just like any other science, your experiment should be replicable. This means someone else can come and test your hypothesis and do the experiment again. In other words, if you find this big black animal, for example, and you think this is a new species, you can write a paper saying this is a new species, right? So how do you test? What if I write this paper, this is a new species, and then Kurt doesn't believe me? And Kurt says, I'm going to check. So what can Kurt do? How can Kurt check? Yeah. So Kurt would go back to this specimen and he would do the same experiments and he would see the results. So in other words, including information in your species description, and I'll talk about this a lot more, but all the information, where did you find your specimen? Where is the specimen now? What museum? What's the museum number? All of these pieces of information make your idea testable. You can test again, okay? So in other words, everyone's like, oh, taxonomy's old and taxonomy's dead and taxonomy's boring. But actually, taxonomy is just the same as any other science. It is a living science, and it is a science that changes over time. In other words, I think this is a species. I described the species. Kurt, 2 years later, comes and says, no, destroys the species. Then Roger comes, says, yes, it's a species. He makes it come alive again. Things can change over time. So just like any other science, our understanding of taxonomy changes over time. In other words, I would not be worried too much about making a mistake, an honest mistake. If you describe a species and you do the best you can, and 5 years later, or 10 years later, or 50 years later, someone says no, that's science. C'est la vie. How's my French? Good, yeah? Good French, yeah? Right? So in other words, in Japan, I think many researchers are worried, very worried about making a mistake. And so they do nothing. There is a sea cucumber species in Ora Bay that is 4 meters long. It's undescribed. We have known about this for 30 years, and no one wants to describe it because there's a species in Thailand that's similar, and everyone's like, oh, I don't know what to do. My advice to you, if you're a taxonomist, at some point, someone has to make a decision, and biodiversity is disappearing. If we wait forever, things are going away. So I think do the best you can, but there is no perfect paper. Okay? So far, okay? Okay, so let's go to the next thing. Describing species. So I'm going to ask all of you, what do you need to describe a species? What kind of data? What kind of information? And there's lots of different ideas. So I'm just going to go around in a circle like a lab meeting. Iori-san, congratulations. We just talked about describing species. So what do you need?
Location.
Location. Okay, so maybe get some information on location. Chloe-san, what else would you need to really do a good job? Describe a species. Specimen. Okay. Okay. So we've got a location. We've got a specimen. Our special guest, Shiori-san, today, for those that don't know, former Mise member, now working at Shirashima Zaidan and also candy supplier and later sticker supplier. So Shiori-san, any idea? Date, collection date. Yeah, sure. Okay. Just write like you read on the label. When? Because things change over time. Gishi, any ideas?
I would start by the basic, like how does it look like?
So external appearance.
Yeah.
Okay, so morphology. Okay.
Basic morphology.
Basic morphology. Okay, good answer. Dr. De Palmas. Information on other species. Okay. We're getting hard. Okay, August.
We need genetic information.
Okay, genetic data. Okay, so we've got morphology now. We've got genetics. Okay. Ooh, Ifena, I'm sorry. It's getting harder and harder. I'm sorry, you skipped the last. Depth. Okay, yeah. Sampling, more sampling to finish. Okay, good escape, Ifena. Mari. Pictures, in situ images would be good, yeah. Of the environment, of the location. Roger. Ecological data, another whole set of data. Okay, cool. Kaidi's trying to hide. I'm sorry, Kaidi. Nandemuyo. Nandemuyo, what do you think? Yeah, to describe a species, Shunokusai no sai. What type of data? For some animals, yes. What does it feel like when you touch it? For some groups of animals, this is actually something important. Each group, each taxon has its own unique characters. For Sunaginchaka, for zoantharians, naide, stinging cells. That's important, right? All right, Sora. Color, sure. So more morphological information combining morphological and photos. Color would be good. Kurt?
I think almost already mentioned.
I know, it's getting really
So I
hard.
think I'll combine niches and sequence idea.
Yeah.
Distinct morphological character compared to.
So unique morphology. What's unique about your species? Okay, okay, cool. Kenta.
気合。
気合
これの種はこれだっていうことを説明するぞ、説明できるぞっていう気合です。
So Kenta's saying, this is this species, and it's not that species, it's this species. Dayo, something like that, right? Like a clear idea about what that species is.
Confidence.
Yes, confidence is important. Jishinを持ってください. And I'm very sorry, Yusuke. Last year.
Collector's name.
Oh, okay, good escape, yes. Collector's name. Who collected or who identified would be good too, right? But a new species. So, okay, all of these things that you said are very, very good for describing a species. My next question to you is, and I'm sorry, online people, I'm not going to ask you, but maybe I've got a chat in there. Gem, holotype specimen, voucher specimen, museum where it is deposited. She's giving us lots of information too. Yes, Gem, you're correct. I'm just going to make sure that now my messenger is going off and making sure there's no excitement today. Okay, it's peaceful, good. Next question. What is the minimum Saiteigen? What is the minimum to describe a species? Let's say you want the ecological information and you want photographs, but no one did that. They just randomly, they put a dredge underwater and they pull a bucket up and, oh, wow, there's something new. So you have no live images. You only know the depth plus minus 100 meters. What? Okay, so the minimum, the bare minimum. No, so okay. So let's put up your hand. Do you need morphology? Okay, so everyone, so bare minimum, saiteigen. これがないとできない. So I'm just gonna write morph. People are saying morphological data. Okay, what else, sorry? Region. So even if you don't know the exact sampling point, somewhere. So yeah, region is fuzzier, right? So, okay. Who thinks region is important? Okay. Anything else? Bare minimum. We've got morphology and somewhere over there. Some picture? Picture? Okay, some people say yes. Picture, put up your hand. Two people, no picture. Sorry, gone. All right. Yes, Gishe.
I would say, like, if you, for example, if you just have a fragment or something, you don't know exactly where, you don't
A
know.
piece.
Yeah, like,
Mhm.
you've lost almost all the data, no pictures. Like, the basic will be, okay, can you try to take it again? So reproduce,
Okay, let's do two things. Number one, do you need a specimen? Who thinks you need a specimen? Okay, and number two, so you're saying you want it to be reproducible.
Yeah, like, if you lost the data, it's like, okay, can we go and collect again?
But then it's a different type. Does your work have to be reproducible? Who thinks yes? Okay, gone. Not enough people. So this is no, we're saying morphology, we're saying an area, and we're saying a specimen. Anything else that you need to describe a species? Okay, of course. Yes. Okay, Muddy wins. Okay, name, you're absolutely correct. We're every single new species needs a name. So do I even write this? Okay, yeah, I'll write this. Okay. But the name is not really part of the data, but it's needed. So Muddy wins. Okay, good job. So anything else that you must have, but minimum must? Yeah, August.
Size.
Size? Do you think you need size? Who thinks size? All right, sorry, size is gone. All right, so the answer, and depending on who you ask, people are going to give you different answers. People are going to say you must have genetic. I think in our lab, you should have genetic because we do a lot of genetics. It would be a waste to not do genetics. But many people say morphology. Hirose Sensei is an expert on morphology. He would say you must have morphology. So depending on who you talk to, which taxonomist you talk to, they're going to give you a different answer on what you need to describe a species. The answer, if I can go back here, is this. Any data or indication thereof. That's it. That is what ICZN says. ICZN does not care what kind of data you have or if it's even good data. It means any data or そのようになりそうな data, any indication thereof. There are crab species erected in the 1950s based on amino acid percentages with no photos and no other information. In other words, ICZN is very, very vague. とっても曖昧. And it's like this on purpose. Not for a mistake, but on purpose. Wazato. The reason ICZN is like this is so that you can describe a species in a very different group and not worry about having to follow specific rules. Every single group of animals is unique and different. And what works in one group might not work in another group. For some groups, maybe DNA is fantastic. For some groups, maybe morphology is fantastic. For ctenophores, Kushikurage, you collect them, they melt. You put them in ethanol, they melt. You get the DNA, great, but the specimen disappears. For Kushikurage, for ctenophores, some people say photographs are enough and you don't need a specimen. And people are still arguing. Do you need a specimen? Maybe no. ICZN doesn't say you must have a specimen. You must have a type, this photograph, this specimen, this nani. But photograph or specimen doesn't matter. That's why people are still arguing. Ja. So in other words, ICZN is incredibly, incredibly vague. But here's the thing. If you want to do a good job, you should include everything that we talked about, right? The more information you have, the more replicable, the more testable your idea is. And the easier it is for other scientists to check and to do more research in the future. So yes, photographs, great. Collector date, yes. Specimen in museum, morphology, color, size, everything is great. Put it all in there. But if you don't have a perfect data set, don't worry. Does that make sense? Yeah? Yeah. When you read ICZN, you're like, what? This is crazy. But this is ICZN. So if someone says, oh, taxonomy, you must have morphological data. That's bullshit. It's not true. You should, but should and must are very different. Okay? Any questions so far?
Yeah, reviewer is gonna say something no.
Yes, so that, but that's okay. That's science. And if you're really, really snarky and picky, if someone says, why don't you have morphology? You could get the morphological data. You need an excuse. Why don't you have morphology? Oh, the ctenophore disappeared. Okay, that's a reason. Oh, I'm lazy. No, that's not a reason, right? So of course, your species must have peer review. Your paper must go through peer review. But peer review is not ICZN. ICZN is different than peer review. ICZN are the rules, peer review is publication. Okay? But good point. Any other questions? Japanese is okay too. Yeah, Chloe-san.
Then who checks if you respect the rule?
So hopefully, and this is another problem, when you submit a paper describing a species, depending on the journal, the reviewer might be a good taxonomist, or they might misunderstand. They might be an ecologist. They might be working with the species, the biological species concept. Biology talks about reproduction and isolation and evolution. That's not taxonomy. Taxonomy is same or different. That's it. So some reviewers might say, oh, you need information on the reproduction. What about their ecology? Blah, 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 blah. And depending on the journal, if you submit to marine biology or coral reefs, most of the reviewers don't even understand taxonomy. They don't even understand CF and AFF. If you submit to Zookeys or Zootaxa or a taxonomic journal, the reviewer will be taxonomic, but they might not give you big comments on ecology that make it easier for your paper to be understood by more people. You need a balance. But it's really clear when you get your reviews back if this person understands taxonomy or not. You still have to answer the reviews, and the editor might not understand taxonomy. So that's why you have to choose your journal carefully. Good question. Any other questions before we move on? No. So this is what I wanted to do today, was destroy your brain. This is like, what? This is super important. Okay, so going back, describing species. What I'm going to do now is I'm going to give you an overview of when you describe a species, kind of the sections, the different parts that you would write. In any species description, there are certain things that you write. Just like if you write a paper, introduction, materials, methods, results, discussion, right? When you do a description, a taxonomic description, there are parts that you must write. Even if you only have amino acid data, there is still information you should write. And that's what I want to talk about today, like a overview. Next class, I'll talk in detail on some of these points, but this is an overview today, okay? So taxonomic account. The first thing you need to write in your species description is a taxonomic account. This means basically, let's see, who thinks they have an undescribed species right now? Oh my goodness, all of you. Wow. Okay, we'll go with Mari. Mari, what's the phylum? Okay, so yes, we're starting easy, Mari. So you would write, like, for example, you'd write Nematoda. Who's the authority for Nematoda? No worries, you can check on worms after, right? But then you write the authority, blah, 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 and the year, blah, 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 blah. What's next? No worries, it's not, I don't want to be mean. Yeah, what's lower? Next, so this is phylum, class. Yeah, oh, do you know the class name?
For the suspected?
Yeah.
Ah, yeah, chromadoria.
So basically, is it chromadoria?
Desmodoride.
So then basically you're going all the way down.
Yeah.
So this is your taxonomic account. And then at the bottom, then you have your, what's the genus? Do you know the genus that you think?
For the one for now, I think it's a new genus actually.
Oh, okay, so we'd have to write the genus. So, okay, Mari, what's the species name?
Huh?
Yeah, just a fake species name. Species name you're never gonna use. Give me a species name.
Bendiella. Longicornis. Short one.
For example, right? Short, very short species name. But basically what you do is the taxonomic account places your species in the taxonomy. And you have the authorities after everything. So no one, for example, Zoantheria, there are two different Zoantherias. One means Hexacoralia, one means Zoanthidae, the order. And so but the authorities are different. So if you write the authority and the year and the names of all of these different taxa from high to low, then it's super clear where you are putting your new species.
According to the previous slide where you said like any data or.
This is not data, this is placing your species.
When you already have
The data we come to that later. So when you describe a species, the first part that you write is the list of where you're placing your species.
Yeah,
This
but
is-
that would depend on the type of data that you have for your species, right?
No, but that's just a hypothesis. That's totally unrelated to this.
Sorry.
No worries, you'll see. I understand what you're saying, but the taxonomic account is where you think your species goes. You explain why after, but the first thing you give is the taxonomic account. Does that make sense?
Right.
Okay, we'll get to the why in a second. Taxonomic account. So you describe the taxonomy from the higher levels downwards, and as well, you might want to list what it is and what it isn't. For example, if you think you have a new species called Mari Bendiella longicada or something like that, okay. And in previous reports, let's say maybe 5 years ago, Shiori-san was working on nematodes, tatoeba, and she described it as SPA1, Mari SPA1. So you could say is same as Mari SPA1 in Kunihiro et al. 2019. So even if other people have seen it, they didn't describe it, but they talked about the same species, you would say, this is it. But maybe Shiori also saw Mari SPA 2, and that's different. You say it's different. Is not Mari SPA 2. So you're listing the taxonomy of what it is, and then if other people have reported it as something else, and is it the same or different? Does that make sense? Some people, yes, Gisha is looking a bit confused because when you describe a species, you give it a name. But some people aren't describing species, they're just doing ecology or molecular or different. And so maybe it has a name like AF or CF are not the species. Maybe it's Mari Af Newtonia, Af Madagascarensis, right? So you say, oh, actually, Shiori said this was Af Madagascarensis, which means I think it's Madagascarensis, but it's not. But here we make it a new species and here's its name. Some people look okay, some people look confused. Basically, you want to tell people, in the past, kakugo de wa kono mono to issho. Kono mono to issho janai. You don't have to write this, especially for maybe Mari Bendiella longicada. Maybe no one's ever seen it before. Maybe no one's ever talked about it. You don't have to do this. But if other people have talked about your species or what you think it is, you should put this in. Okay? これマストではない。誰か何か報告があるなら。Okay? こんなになりそうなら put it in. Okay? Okay, cool. So, after that, the next part that you would list, this is exactly like a paper, material examined. Here you list what you examined, whether it's a specimen or whether it's a photograph. And so that in here, you would include all of those things like list it all, location, date, depth, region, collector, blah, 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 all of that information, museum number, all of that goes in there, okay? As well, if you have many, many specimens, you need to choose one. That's your type. You need to choose one specimen that's your type. I'm going to talk about this in another class because there's a lot of information in here. But for now, you choose what you think is exactly what Kenta said. Actually, when we asked everyone what you need, yours was the best answer. この species だよ. That's all you need to describe a species, right? So, but when you choose a type, if you have many, many specimens, you choose based on your data and your thinking and your observations, you choose what you think is the most representative specimen or the best specimen or the most complete or whatever. One specimen, that's your type. Okay, so for example, holotype. The number one is the one is the holotype. You could choose a couple paratypes. Paratypes are like backup. Maybe you put the holotype in a museum and a tsunami destroys the museum. Then the paratype can become the holotype. So these are like your backup, your extra, like vice president. ですよね?ですよね? Right? Okay, kind of. And also the locality. So where the holotype was collected, if you found the holotype in Nakagusuku Bay, the type locality is Nakagusuku Bay for this entire species. This species is from Nakagusuku Bay. I'm going to talk a lot more about this in a whole nother class by itself. But for now, when you talk about material examined, you list all of your material and you say, this is the holotype. This is a paratype. Okay. Sometimes for some papers, material examined is like 10, 12 pages, hundreds of specimens. It happens. Sometimes it's only one. I've described species on one specimen, and I've described species on hundreds of specimens. It's case by case. Okay? Yeah.
If you look at some other species that were previously described.
That's coming up. Don't worry.
Okay.
It's just like Gishe. It's like, what do we have? Patience. No, don't be sorry. It's good. Can't a question or just fixing hair? Kayu desu ne. Okay. Then the next part is what's called the diagnosis or the differential diagnosis. This is not the description. Could be one, could be two, could be three sentences. This is how you say your species is different from every other species, especially close species. So if you find Bendiella longicada, and you say, it looks like Madagascarensis, but it has blah, 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 or it is blah, blah, blah. Yeah.
AKA delineation.
Maybe some people, usually we call it diagnosis. Delineating is just a general verb for separating. It's also used in molecular phylogeny. Diagnosis is a specific taxonomic account to say it's different, but close. Some people might have written delineation in some groups, but as far as the groups that I've worked on, which are amphipods, octocorals, zooantherians, It's usually been diagnosis. It's always been diagnosis. This, but basically what it is and how it's different, how it is unique. This is the part where you say, this species is, you can identify it by amino acids, any data, anything, whatever data you have that shows that it's different. That's where you put that. Sometimes some people mix this with the next part, which is the description. The description is all of this information. This is where you put all your data, right? What does it look like? I mean, you don't have to. Anything in here is okay, right? Any data or indication thereof. But you put maybe external, maybe you put internal, maybe you put other ecological, phylogeny, naidae, lots of figures, everything goes in here. Some people combine the diagnosis and the description, and the diagnostic part, the unique part, they make in bold. 太文字で、これが unique. So there's not, the diagnosis and the description are often mixed, but you can put them separate too. Whatever's easier, right? But this is where, so when you describe a species, you're doing two things. Number one, you were saying, or you're doing three things. Number one, you give it a name, right? You give it a name. Number two, you say it is different from everything else by this. Number three, you say, and this is what it is. This part says, this is what this is. This part says, this is unique. And the taxonomic part says, this is the name. Does that make sense? Question?
Yes, so for the description, this is where you look at, for example, if I gather a team and say like, okay, let's go to Amami Jima to I know that there is an iguana, right? So everybody will read this and try to look for this character, right?
Yeah. Yeah. But you could, much of the information about whatever animal you describe is the same as other animals that are closely related. You still have to put it in there, right? There are many things are similar about many species. If we are going to describe a chimpanzee, much of a chimpanzee is same to a human. But we still have to put 2 arms, 2 legs, 2 eyes, right? All of that goes in here. You don't have to, but some as much as you have, right? Question, anything else? So far, so good? Okay. Lots of figures. So also in here, if you have in situ figures, if you have, we talked yesterday from Chloe's talk about drawing, you can put drawings in here. Lots of groups, especially Crustacea and also probably polychaetes, right? Lots of people do drawings, photographs, cross sections, histology, anything can go in here. All of this can go in here. Okay. Then some people put a section called distribution. You don't have to, but if you have a lot of specimens, you could put where you found it. If you have specimens from different locations, your one specimen is your type. That's your type locality, but maybe you find it in many places. So you could talk about there. It's not needed, but it's helpful. However, you cannot make a new species only on location because location is not about the animal. When you describe an animal, you have to describe the features of the animal, not the location. Does that make sense? Some people are, 簡単に言うと、種を記載する際、どこで見つけることだけで種の記載できない。場所が種、その標本と関係ない。標本に基づくデータしかできない。記載。So if you go to Galapagos and you find a zoanthera and you say, oh, this is a new species because no one's ever saw one in the Galapagos, you're wrong. You have to compare the animals, not the location. Okay? This is something some people also forget. If you go somewhere new, very far away, the Galapagos maybe, there's a very high chance what you find is new, but you need to look at the specimen, not at the location. Then the last section is fantastic. This is called the remarks or the notes. And this is, oh, this isn't the last, but it's almost the last. And in here, you can put anything else. Anything else that you think is directly important to talking about your species. You could talk, maybe you think it spawns in June, like Gishe with Nanipora. We have a little bit of information, maybe it spawns in summer. Okay, cool, put it in there. You could say, oh, it's found in cracks in these caves. Okay, you put ecological information here. Okay, cool. Our molecular phylogeny actually shows 2 groups. In the future, maybe this will become 2 species. That's cool too. Anything and everything that you have, you can put it that doesn't fit in other sections, you can put in here. Whoa. Okay. Then we get to the part that Maddie mentioned first, etymology. I'm going to talk about this in another class as well. This means the name. This is where does the name come from? What does the name mean? What language? If it's Latin, is it masculine, feminine, neutral, neuter? So this is where you explain why you are giving the species this name. Have fun. Some scientists are very, very serious and only use Latin or Greek. That's fine. That's traditional. That's cool. Takuma Fuji, for example, my first student is the complete opposite of me. He's very serious about his names. I'm an idiot. Nanipora, right? But my thinking is the name should have meaning to you. The name should have, even though the animal doesn't care. Well, maybe it does. I don't know if that's bad, but the animal, you could give it any name, right? But I usually, if I'm in Japan, I like to give animals Japanese names. I like to give animals names that are easy to remember. Usually, Antipathozoanthus, I'm sorry, very long name. But to me, it's easy to remember because there's Zoanthus on Antipatharians. What's a genus name? But this is your one chance. The person who discovered this species gets to choose the name. So this is really cool. You could name it after your sensei. You could name it after Shiori for fantastic snacks. Anything's okay. The only thing you shouldn't do, don't name it after yourself. You can, there's no rules, but it's really, really cheesy. It's like Donald Trump. Donald Trump would do this. Okay? But generally, don't be Donald Trump, okay? I don't care about politics, but in taxonomy, don't name it after yourself. Yes?
Is this a rule or something that you said?
This is my advice to you. This is also not a rule. Having fun is not a rule. You don't have to have fun. You can be very serious. Like for this is not ICZN, but like the genera of Zooxanthellae of Symbadinaceae used to be clades A, B, C, D, E, F, G. We gave them those names on purpose. Symbadineme is the original, it stays. B is Breviolium. C is Cladocopium. D is Duristinium. E is Ephrenium. We did B, C, D, E so people could remember. The names are very serious and very Latin, but we had some fun and we made it easy to remember. So when you come up with names, have fun. We'll talk about this in another class, but there's a lot of checking, a lot of things you need to do to make sure that your name is okay. But we'll talk about that in another class. Yeah.
Is it found in the ICCN, the suffix like II, AY?
So no, but basically you have to follow the rules of grammar for whatever language you're using.
Because
We're going to talk about this in another class. Huh?
If it's location or if it's name, like.
Yeah, we're gonna talk about that in another class. There's a lot, and there's more than just Latin and Greek. There's a lot of different rules in there, and there's also invalid names and non-available names. There's a lot of stuff in here, so it's gonna be its own class on names. But for now, this is Mari.
There's like strong differences between the female and the male.
Then you have to describe them both if you can, depending on the taxon. Maybe people say, oh, you only describe the male, you only describe the female. Each group has its own rules. So in Crustacea, many people say, oh, we can't describe it because we haven't found the male. But that's a Crustacea rule. That's not an ICZN rule. You could do whatever you want, but you're going to have to go through reviewers.
But okay, you still have to designate your type. So
Yeah.
there must be one. When there are huge differences between.
So you could
You describe the male.
it's taxon to taxon. So like maybe in some groups, it's always the female, in some groups, it's always the male, in some groups, no one cares. And you'll have to read the other papers and see what they did, and then see if it's a rule or not. And if it's a rule, but you think it's crazy, like maybe there's 99% male, like some of the sea turtles now, or it's 97% female. You can try to change the rules, but you still have to go through peer review. Does that make sense? Sorry, it's not a great answer, but it's, yeah. Yeah, okay. Anything else? Okay, points to consider. This is the last slide, just some things to think about, right? All species are hypotheses, right? We talked about this at the beginning, but I really want to stress this. Don't worry if you make a mistake. Things happen. It's okay to be wrong. I just said that. Consider your types carefully. This is something I want to talk a little about. I'm going to talk more about the types and the rules and the types of types in another class. But here, what I'm trying to say is, if you have a lot of data, let's say you have 10 specimens, 10 Peronia specimens, and you have the DNA, and you think, oh, this is a new or undescribed species, okay? But you actually look at the DNA, and there's a little difference. You have 5 in one group, like you have Nanipora, and you have 5 in another group, and there's like one single base pair difference. And you're sure, you know, you have at least one species. But maybe there's 2. You don't know. You can just do one species, and you can put in the remarks, maybe in the future, this will be 2 species. That's okay. But your types should be all from one group. If you mix your types across the different groups, when you split, you create a problem. Which name do I put on which group? I don't know. Yeah, so
This is actually like the big problem right now in Avipatorium.
it's a problem in Zoantharians too.
The type specimen of the order is in the wrong family.
Yeah. Well, and originally it was an octocoral, it was a Gorgonian, right, or something. Yeah. So you have all sorts of problems. So when you're designating your types, choose the best type. But then the other paratypes, or the ones, make sure they're the closest with whatever data you have, and don't choose something from another group if you have groups inside your species, because you are far away, right? Oh, I've got one from Japan and one from Ethiopia, like, oh no, bad news. So we'll talk more about types later, but this is just advice, especially some groups might have cryptic species, right? Okay. ちょっと待って。
So I feel like so you want to give as much information as possible, but at the same time for your type, you want to be
specific, because we're not talking about a biological species. Your description is of the type specimen usually.
Yeah, but I mean, if you have the information for the same species that you're describing, but for example, it's in the Galapagos. But you have that information, so you can put it in the distribution and all of
Or you
that.
can put it in the description, you can say specimens from the Galapagos have blah, 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 blah. Or you could put it in the remarks, specimens from the Galapagos were a little bit different because they had blah, 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 blah.
So your specimen for the Galapagos, you don't consider them paratype. They're like additional material.
Additional specimens. They're still inside the species. If you think maybe there's 2 or 3 or 4 species, your types should all be from one group to prevent future confusion, just as Kurt said. In some groups, this is super bad. It's super hard and people don't know what to do. Sometimes people will just throw all the old names away and start again because they don't know what to do. Or other people just say, oh, I don't know what to do, and then no one does anything. It stops taxonomy. Okay?
Okay.
The other thing is, this is the last one here, this remarks section, the notes section, that last section before etymology. That can be useful because you can put your ideas or your thoughts or your maybes, moshikashite, in here, and you can protect yourself.
Disclaimer.
maybe this is 3 species, but right now we're only making 1. It's a way to sort of to hedge your bet, so to speak. So the remarks section is super useful, and often I use it because you don't know, right? Any questions? Anything online? Any questions? Everything? Everything? Life? No, yeah, anything in today's class.
No, I have the question last.
Last class, yeah, okay.
I was watching into, you're explaining that ICZN, you said that, only up to class. In the first slide, the very first slide.
No,
I-
you correct, like,
Yeah.
I corrected myself. It's actually only to
To
family,
family, up to family plus.
up to family, no, class is higher.
Yeah.
So I made a mistake in my slide and I corrected myself verbally maybe, but this is where it is.
Okay.
Order. I'll double check, but I think it's here. Okay, okay. So, but generally, usually we all, like, even though ICZN is only up to here, we usually put the authorities for everything, so that it's super, super clear. So we try to follow these rules here, but we don't have to. It's not, and I'm gonna talk about this, the rules more when I talk about types and names. Because in the end, if you think about it, ICZN, international, okay, kokusai, code, rules, zoological, dobutsu, dobutsu gakuteki, nomenclature, names. ICZN really is governing names more than anything. They're not governing data. They're not governing hypotheses. And so we need to have a class on names and we need to have a class on types. Yes, and there's a few other things too. Anything else?
How to become a member of them.
I don't know. I've kind of been a member in the past. I can't remember. But if you really want to be, you can, but you spend a lot of time arguing over a lot of small detailed stuff. For example, there are 40 different names for the African elephant. And there are groups of taxonomists arguing over which name is the valid, correct name. People have been arguing for over 100 years about what we should call an elephant. And it's super, and people are super, like really aggressive about it, right? So I'm a taxonomist, I give species names, but I don't care what you call it as long as we can all agree. At the same time, I do not think mistakes are good. So I think there's so much diversity on coral reefs. We're not going to be perfect, but we need to do the best we can. That's sort of my thinking on it. Elephants. All right, anything else? Chloe-san.
Right, so,
Chotto.
in the description, so what do you describe? Like, do you describe the holotype or do you put all the information?
There's no rules on that, but I usually describe the holotype in detail. And I might describe if there are some other specimens that are a bit different. I might say the holotype looks like blah, 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 blah. It has blah, 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 blah. It has blah, blah, blah. We looked at the Naidae in 3 specimens, and this is what we saw. So it doesn't matter if you're describing only the holotype or multiple specimens, but you should note anything that's different or unique among all the material you examined. But there's no hard and fast rule about this.
Because like for the
Some people will they'll split description. Let's say description of the holotype, descriptions of others. You can make subsections in there as you like. You could put Naidae, molecular phylogeny, external appearance, internal appearance, as you like, and then list each specimen and what you did. You can put tables in there too. Anything's okay. Tables, figures, anything.
I feel like some description I've read, it really looks like a meta description and it's hard to understand. Like, for example, they'll say, oh, this worm is between 40 and 50 segments. But then there is not information of how long is the holotype.
Yeah. So but that's not wrong because ICZN doesn't care. It sucks for you following and trying to do the science. So if you think about, okay, this description is missing this information that I would like, then as a scientist, you should try to get that information for the next generation, right? Yeah. No, so when you're reading a description and you're running into problems like, wow, I don't know, I wish, remember that when you publish your paper, if that makes sense? Okay, cool. I mean, especially old papers, very old ICs, very old papers, 1 line, 2 lines, one hand drawing. Like really tough to, what do you do? We'll talk about that in another class. Super fun. Okay. Okay. Let's take a 2-3 minute break. And let's take a 5-minute break. You guys can eat some snacks, check your makeup, and then we'll have a really quick lab meeting and then we're done. The lab meeting is very short today. Okay. Thanks everyone online. I'll keep you guys online for the lab meeting, but I'll stop recording.